presentation will be recorded for future reference. And yeah, I'll give you guys a few minutes before we get started with introductions. For any attendees that just came, again, I just wanted to say that my name is Alpha Gasama, and this is Dr. Karlsson, and we'll be waiting a few minutes before we get started with the introduction and presentation, just so everybody's situated and things are settled. And I also wanted to let you guys know that this will be recorded in case you know it needs to be used for future reference by any of you guys. So, sit tight. Just wait about 2 more minutes, and then we will get started. Still 10 attendees. Wait 1 more minute, see if anyone else can join us beforehand. Hopefully everybody's having a great Wednesday, enjoying their quarantine. Well, or taking a walk if you can. I didn't walk today, but I usually do.
Not at this time of the day though, Alpha.
Oh, really?
Too hot right now.
Oh, okay. See, I didn't go out today, so I don't know.
I like to go out in the morning and then in the afternoon. I will, or in the evening. Sometimes I will get out in the morning, or in the middle of the afternoon when it's hot, but I usually regret it.
I usually go in the morning before it gets too hot. Okay, it's 2:03. So let's go ahead and get started. Again, just to reiterate that this is a recorded session. So for future reference, if you guys would like to refer to it, it will be there. I guess Dr. Karlsson can start herself off in introductions, and I'll go after.
Yep. Hi, I'm Amy Karlsson. I'm an associate professor in chemical and biomolecular engineering. And my research involves protein engineering. You'll hear a little bit more about that when I talk about my work. And I'm specifically interested in using engineered proteins and peptides to study and potentially treat human disease. I also thought I could give you one of my favorite things about Maryland, and that's really that I really love the people at Maryland. It's really fun to work with students and other faculty that are just so smart and so excited to learn. And also, I guess, what could be better than a mascot that's a turtle? You know, I don't know anywhere else that has a turtle as a mascot, so I like Testudo.
Cool. And I am Alpha Gasama. I'll be helping moderating this event. I'm a rising senior, and I'm also a chemical engineering major. So I actually had Dr. Karlsson last semester, so we know each other pretty well. And my favorite thing on campus, this was a tough one, but I would have to say how beautiful it is. I really enjoy being on campus, seeing McKeldin Mall and the fountain. It's really beautiful, and it's a really big campus, so there's a lot to explore. So I would say it's really tough. There's a lot of options, but that would be my favorite. Okay, so now that the introductions are settled, without further ado, I'll let Dr. Karlsson get right into her presentation.
Okay, so get my screen shared for you.
Oh, I'm sorry. Before you begin, Dr. Karlsson, I just want to mention, if you guys have any questions, feel free to ask in the Q&A. We encourage you guys to ask in the Q&A, but if you ask in the chat, that is fine. It's possible that it might be answered later. Okay, sorry about that. Go ahead, Dr. Karlsson.
No problem, that's important. I like to answer questions, so put them out there for us. And Alpha and I can both answer questions. and about whatever comes up. So before I get into really talking about my research, I thought it might be a good idea to give you a little bit of an introduction to chemical engineering more generally. And what is chemical and biomolecular engineering? Because some of you may not be familiar with what that is. And chemical and biomolecular engineering is really the application of science, math, and economics to convert raw materials into more useful or valuable forms. So pretty much anything that has to get manufactured, you can guess that a chemical engineer is probably involved at some point. And I've put below some pictures of things that chemical engineers work on, things like drugs, fuels, so ways to make your car run, whether that's from oil or whether that's from batteries. And these are actually a couple things I worked on when I was an undergraduate student on my internships. I worked on masking tape and I worked on biomedical electrodes. I have friends who worked with at General Mills and worked on all different kinds of food products, including fruit roll-ups, which is kind of interesting. And one of them told me you would be really shocked at how much chemical engineering goes into making fruit roll-ups. Also, things like detergents and Tide Pods. My husband actually, for one of his internships, he's a chemical engineer by training as well, and he worked on Tide Pods when they were something new. So in addition to really thinking about making these products, we also think about really engineering molecules, and that includes biomolecules. And that's why we have biomolecular engineering in our name, to really highlight the fact that we do work on things that are important in biology. And when we're trying to engineer molecules, we're often working on these same types of applications. We're looking at drugs, fuels, plastics, food, batteries, and a lot of other things. And just to also give you an idea of what specifically goes on in our department at the University of Maryland, I want to highlight a few different projects that have gone on in our department or are currently going on. The first is something that was actually developed by two students who were undergraduates and graduate students in our department. And they worked in Srini Ranavon's lab on polymers, and they were working with this really sticky polymer. And what they decided to do was turn that sticky polymer into a product. So they've actually created a product called Grip Boost, where you can add it to your old football gloves or any gloves for any other sports and re-get the sticky into them. I didn't realize that football players had gloves to help them catch the ball that were sticky, but that was new to me, but that is something that's important. And this lets you reuse gloves and not have to buy new gloves as often. Another area of research that's in our department is in materials for batteries. And this is work in Chuncheng's Wang's lab. And they're really trying to make batteries safer and better so that we can use them for all the new types of applications that we're working on that need batteries. Another area of research that's really interesting is from Akuya Sayawuku's lab, and her lab works on aerosols, so things that get into the air. And she's really interested in looking at cloud formation and how do clouds form, and also the other chemicals that might be in the air that might cause pollution and any other kinds of issues in the air. She's also interested in looking at how aerosols affect health. So there's been a lot of talk around COVID-19 and whether it gets into the air, whether it's in aerosols. And so that's the kind of thing that she might be interested in studying. And the last one that I want to highlight for now is work of Jeff Clauda's lab. And his lab does something that I find really interesting. And they look at everything on the molecular level, maybe even on the atom level. So his group does molecular modeling to try to understand what's happening on that really small level that we can't actually see. So his lab is particularly interested in looking at the interaction of proteins, which I'll talk about when I get into my work, with cells and the kind of the membrane that surrounds cells. So you can see we do a wide variety of things in chemical engineering, and I think that's one of the things that attracts a lot of us to the field, is that we can take our knowledge and apply it in a lot of different areas. What I'm going to talk about today is the specific work that's going on in my research lab. And in my research lab, we're focused on protein engineering, and specifically on engineering proteins and peptides to target a fungal pathogen. the first thing that I want to talk about is, what is a peptide? For some of you, this might be a new term, or at least it might be something you've heard. Sometimes you hear it in commercials. They talk about peptides in shampoos and conditioners all the time, but they don't really tell you what they are. And peptides are really just a short chain of amino acids. So the structure that I've shown here is an amino acid where we have a nitrogen on this side. This gives us our amine group, and a carboxyl group on the other side that gives us our acid. So we have amino, acid to make the amino acid. And in this chemical structure, there's also a group that can change. And there are a lot of different structures that can go where I've put this R here. And you don't need to know the details of all of these, but I put up a figure on the right that just shows the diversity of the different structures that you can see. And if you know chemistry, those different structures will mean something to you. If you don't know chemistry, what's important is to look at the fact that they're all very different. And some of them, for example, have these rings in them. Some of them have a lot of just carbons, and some have some nitrogen groups. Some of them have charges. So we might see positive charges on our side chains or negative charges on our side chains. And so those are really important to how those amino acids behave, and also to how they behave when they're in a chain that helps make up our peptides. So a peptide is really a bunch of amino acids connected together. And I've shown just 3 here, and I've highlighted how each of them is different and how each of these amino acids could have a different one of these side chains from what I've drawn on the right. And these are natural molecules. A lot of things in your body are made up of these types of structures. So peptides are short. And that's important because sometimes we can also think of a peptide as really just a small protein. So if you're familiar with what proteins are, they're large molecules. These are really, really big molecules, and they have a lot of complex functions in living things that really allow us to live and allow our bodies and to do the things that they do, all of the functions that have to happen on a daily basis for us to live or to play sports or whatever we're trying to do. And peptides really have similar types of functions. They're just a lot smaller. So whereas a protein can be on the order of 400, 500, 600 amino acids, maybe even bigger, peptides are usually on the order of less than 100 amino acids. So they're substantially smaller. And the ones that my lab works with are actually usually less than 50 amino acids. So I just mentioned that nature has a lot of proteins and peptides, but I said that my lab wants to engineer them. So why are we engineering these when nature has already given us so many of them to start with? One of the main reasons we want to do that is because we're now trying to use proteins and peptides in new places and for new functions. So for example, a lot of the proteins in nature, for example, the ones in our bodies, they've been evolved by nature to function in the conditions in our body. But we might want to take them out and have them function in someplace new. Maybe we want to use that protein or peptide in an industrial process where it has to function at a temperature much hotter than what's in our body. So in order to do some of these new things, we need to think about 2 different aspects that we can engineer. First, we might want to improve the ability of a protein or peptide to function in a new environment, because as I said, we're using these things in new environments. So we might want to increase their thermal stability. We might want to increase their stability in the presence of proteases, which are molecules that like to cut up proteins into small pieces so they can't work anymore. We also might want to improve their solubility, so how much of it we can put into water, for example. And that can be really important when we want to produce a lot of a protein, more than just to study in the lab, but if we want to produce enough that we can sell it to the world, it's important that it's really soluble to make that process easier. A second area that's important in protein engineering is thinking about improving the function of a protein or peptide, so improving on its natural function, or actually introducing a brand new function. So we can do things like improve or expand the enzymatic activity. And I've shown an example on the top right where there's a chemical reaction where it used to use a heavy metal catalyst. So something that wasn't particularly environmentally friendly to complete the synthesis of this drug. And instead, they decided to use an enzyme, which is a type of protein. And by doing that, they were able to really make the process a lot more environmentally friendly, and also to improve the process and produce better product than they were getting before. Another thing that we can do is we can think about trying to make things have strong antimicrobial activity. And this is something that my lab really likes to work on. And it's something I've been working on for a long time. So this is just an image of a molecule that I've worked on since I was a graduate student. Additionally, we might think about trying to make things that bind better, that have a higher affinity. They like another molecule more than they do naturally. So I've shown one here where we're looking at, for example, an antibody. This is just a small part of an antibody binding to a target in the body. And this antibody is actually used as a treatment for an eye disease, for macular degeneration. But you could also imagine the same type of molecule, an antibody, might be something right now that a lot of people are working on to try to engineer to target the COVID-19 virus, to try to think of it as either a treatment or as a preventative measure for COVID-19. So this is a place where there are a lot of protein engineers that are working. Not me right now, but it is a really big place for protein engineers. So in my lab, when we're engineering these peptides and proteins, we're really trying to use them to enhance our understanding of cells and of disease. So I'm very much focused on problems in medicine. When I was in high school and actually even earlier, I really wanted to go to medical school. I thought I was going to be a medical doctor, and it turns out I actually like engineering a lot more, but I still haven't given up my love of biology and of medicine. So most of the applications in my lab are focused on ways that we can use engineering in medicine. And we're trying to design proteins and peptides that we might be able to use as research tools, for example, to study disease. We're trying to design things that might help us detect diseases or identify the cause of a disease. And we're also working a lot on things that would help us design new medicines to treat disease. And that's what I'm going to talk about today. Most of the projects in our lab right now are focused on fungal disease, specifically a type of fungal disease called candidiasis. This is probably not something most of you are familiar with, which is good because it means you haven't had to deal with this disease. But fungal infections caused by Candida species are actually really important infections in medicine. And one of the reasons is that Candida species of fungi are actually commensal organisms. This means that they live in our body, and probably all of you listening to this webinar right now have this in your body. It's probably in your mouth, it's probably in your GI tract, and normally it doesn't cause any kind of disease, and that's why we call it a commensal organism. However, if your immune system is compromised, so for example, you're sick and your body isn't able to fight infections as well, the organisms that are in your body can take advantage of that opportunity to cause disease. And that's why we call them opportunistic pathogens. They're really just, taking they see that opportunity and they're going to jump in and cause disease. And they can cause a lot of different kinds of diseases. And I'm showing a couple pictures here to help you see. The one on the left is a picture of a baby that has a skin infection that's caused by a Candida species. So this is something you'll see a lot in infants. Diaper rash can actually commonly be caused by these pathogens. The image in the middle it's one of my favorites. Hopefully people aren't too grossed out by this, but I think it's a really interesting image because that's actually an oral fungal infection. That white stuff on this person's tongue is yeast growing on their tongue. And that's because, as I said, this pathogen can be in your mouth. And normally it doesn't cause disease unless something happens. So for example, if you have a person who has AIDS or a person who has cancer and is undergoing chemotherapy to try to get rid of the cancer, it also can get rid of some of their healthy cells that help us fight infection. And those people are very susceptible to getting oral thrush, as are infants as well, particularly newborns. So this is something that also, if there are any parents out there, you may have seen in your own kids. In addition to these diseases that are kind of a little bit more on the outside, these same organisms can cause bloodstream infections that can be really expensive and really, really hard to treat. So, and very severe as well, with mortality rates that are about 50%. So that's a pretty high incidence of death from these diseases. When we're thinking about treating diseases caused by these Candida species, we have to think about the most common species that we're going to see. And in the US right now, that's Candida albicans. And there's a nice image here on the right that I like to show people because it shows the fun biology of this pathogen. So I guess most people don't think of yeast infections and pathogens as fun, but to me, they're really fascinating. And if we look at this organism, we can see kind of these round cells. And there's lots of these little round cells in this image. And that's kind of the typical yeast form of this organism. So this organism is really closely related to the yeast that you use to bake bread. So it looks a lot like that organism. It's a little bit smaller, but it is closely related. But interestingly, under certain conditions, this fungus can also grow these really long structures called hyphae. So these are actually this is the identical organism here. This isn't different organisms that make these different cell shapes. It's the same organism, and just under different conditions, it'll grow differently. So that's one of the reasons I really like to study Candida albicans, is because it's got a lot of these kind of fascinating biological features that some other organisms don't have. And when we're trying to treat these infections, there really are a lot of problems associated with that, because sometimes these organisms are resistant to drugs, so they just don't respond to our treatments, or as we treat them, the diseases, they develop resistance to the drugs. In addition, at least one of our really good drugs to treat disease is toxic to humans. So we have to do some special things to try to get humans to be able to tolerate the drug without getting sick from the treatment itself. In addition, Candida species will also grow on devices, medical devices. So things like pacemakers, knee implants, dentures. So anything that you put in the body, this can grow on and cause a really hard to get rid of type of infection. So because of that, we're really interested in thinking about new ways that we can treat these infections and come up with new drugs that we haven't used in the past. And I'm going to talk today about two things that we're doing that kind of will help us move towards new treatments. And the first one that I'm going to talk about is our work to design some antifungal peptides. And I've shown the images here of the people who have worked on this project in the past and who are currently working on it, because I think it's important for you to see that these are real people that are doing this. So these are some graduate students from my lab that have done this work. So let's start by just talking about the peptide we're working with. This is a peptide that we get from nature. And as you can see in the title of my slide, this is actually a peptide that we get from your spit. So you have this peptide in your mouth, in your saliva, and it naturally helps keep any of that fungus that might be in your mouth at a low level so it can't cause disease. The structure of this peptide is shown here. These are just letters that represent the different amino acids. The specific amino acids aren't important, but the ones that I've highlighted are interesting because they all have a positive charge. And so that gives this peptide a really high level of charge that's important for its activity. We don't know exactly how histatin 5 actually kills Candida albicans, but we do know that it has to get inside the cell and interact with things inside the cell in order for it to kill the Candida albicans cells. So this is great. You know, we do have some Candida in our mouth, but we have a peptide that helps keep that Candida in check. Unfortunately, the Candida albicans also has a weapon of its own, as one of my graduate students like to say. Candida albicans produce proteins that are called secreted aspartate proteases, or SAPs for short. And these proteases are something that help the Candida albicans cells cause disease. And there are actually 10 different proteases produced by the cells. And these proteases can cut proteins and peptides at positively charged residues. And remember, I just said that this peptide is full of positively charged amino acids. And previous work showed that these weapons of the fungus actually can cut up that peptide in our mouth and make it ineffective. So what my lab wanted to do was to see, could we think about trying to make this natural peptide better? So we know the natural peptide can be cut into small pieces by the enzymes that the fungus produces. And when that happens, we now have weak antifungal activity. These small pieces aren't able to kill the cells like the intact peptide is. So what my lab wanted to do is we wanted to introduce changes into this peptide so that we could block the action of those proteins produced by the fungus. And I like to think of this as kind of making my ninja peptide that's going to fight off the fungus. And so you can see that its nunchuck right here is it's getting in that scissor, so that scissor isn't going to work very well. And so if that peptide can't be cut up by the fungus, it's going to stay intact and it's going to maintain its strong antifungal activity. So what I'm going to tell you about is a little bit of what we did and some of our results. What we did was we took a few of those amino acids that had a specific kind of side chain. So these were the ones that had this lysine or this K at specific locations in the peptide. You can see there's 4 of them. And we made changes at each of those locations where there was a lysine. And we changed it to 2 different amino acids. We changed it to an arginine, which also has a positive charge, like the lysine. And we changed it to a leucine, which doesn't have any charge. So it's a neutral amino acid. And we looked at whether these peptides could maintain their antifungal activity. Because although our main goal was to try to make the peptide be resistant to getting cut up, we also wanted to make sure it would still maintain its ability to kill the fungal cells. And what was the good news for us was that the peptides, before they had interacted with the proteases that cut them up, they were able to all maintain some level of antifungal activity. The black is the peptide before we made changes, and you can see some are a little bit better. So a higher signal on this graph indicates a stronger activity against the cell, so it kills more of the cells. And below the black, it's a little bit lower. It doesn't kill it quite as well. But the good news is all of them still killed to a good extent. So then we decided, let's look and see, how did our changes affect the ability of those proteases, our little scissors, to cut up the peptide? And we did this by mixing our peptide with those different scissors. So in this case, we're looking at a specific one called SAP9. And if we look at our peptide that hasn't been mixed with any of the SAPs, any of our scissors, we only see 1 band here. And that indicates that the peptide is still intact. All of the peptide is still in one piece. But if we take our natural peptide, histatin 5, and we mix it with our SAP9, we see in this second lane, we see 2 different bands. So now our peptide has been cut up. So only part of our peptide remains intact, and some of it in this lower band has been cut up. About 50% of it has been cut up. So we've quantified the results from the gel above to tell us how much of our peptide remains intact. And we looked at all of our different variants in the same way. And what was exciting for us is we found 2 variants, this one we call K17R and this one we call K17L, where we see a lot more of our peptide is intact. So we can see it in both this image and this image, along with in our plot, we see about 80% of our peptide remains intact compared to the natural peptide that only has 50% intact. So this was really good news because we succeeded in creating a peptide that was more resistant to being cut up. Interestingly, I think this is really important to point out because sometimes our experiments don't go the way we plan. One of our other peptides that with the changes that we made, we thought this might help. But if you look at this peptide, we go to having 0% of our peptide that's intact. So all of this peptide has now been degraded. So it turns out that this was a bad guess on our part. But what's important is now we know some things that make the peptide better, and we also know some things that make the peptide worse, which helps us start to understand what are kind of the rules for how this peptide interacts with those little molecular scissors that cut it up. So now maybe we can start to understand even better how to design improved peptides. And real quickly, I also want to show you the antifungal activity after we cut up these peptides. So we mixed our peptides with our little molecular scissors. And so in some cases, we have a lot of peptide that's still remaining intact. And in some cases, we have a lot of fragments. But it's possible that some of those fragments might still be able to kill the fungal cells. We may not need the whole peptide. Maybe we only need a small part of the peptide. So on this image, what I'm showing you is at different concentrations of a starting amount of peptide, after we've cut them up, how much antifungal activity do we have? So we want to see a high level if we have a good peptide. So this gray line shows what happens to our natural peptide when it hasn't been degraded. So we see a lot of activity. But after our natural peptide has been degraded, in this dashed gray line, you see a lot lower activity. There's a lot of the cells are still alive in this case, where they're all dead up here with the gray. And the good news is 3 of our peptides resulted in a big improvement in antifungal activity following degradation. And 2 of those peptides, we think that is because they didn't get cut up. And 1 of those peptides, we think it's because our changes actually changed how it got cut up. And so even though it still got cut up, it maintained its antifungal activity. So the next thing that we did was say, okay, well, we've got some good peptides here. And we only have 1 change in each of our peptide. So what happens if we combine 1 change from each of our peptide into a new peptide? Can we make it even better? So what we did was we combined the K11 modification along with our K17 modification. And what we found was that we made our peptides even more resistant to getting cut up by that molecular scissors. And we have actually about 90% of our peptide remaining rather than about 40% of our histatin 5. Or with this other SAP2, we can't even detect any degradation. As far as we can tell, under these conditions, there's no peptide that's been degraded. So that's really exciting. And additionally, we can see the effect of the peptide staying intact in our antifungal activity assay, where we see really high signals for how many cells we're killing. So we've reduced the viability of our cells a lot, and even better than the original peptide that we would get from our saliva. So we've started with something from nature, and we've actually improved on the design from nature by using some of our skills to think about what was happening and how that peptide was interacting with the cells themselves and also with the molecules produced by the cells. And something that we did that we also thought was really interesting is we took our peptide and we put it into a surface coating. And the idea was maybe we could prevent some of these infections that are caused by the growth of this fungus on a surface. So for example, let's think about somebody who might get an oral infection or is really susceptible to getting that oral infection on their tongue. You could think about giving them a mouth guard, for example, something to put over their teeth. And you could coat it with the peptide and it would constantly release a little bit of the peptide and prevent those people from getting this infection. And what we found was if we look at a surface that had no peptide or a surface that had the natural peptide in it, we still see a lot of growth on the surface. But if we put our modified peptide, the K11R, K17R peptide, which I put my ninja up here, it's my ninja peptide. If we put our ninja peptide into our surface, we see very little growth on the surface. So we're really excited about these results because it's kind of taking some of this knowledge and this design from just studying the molecular interactions to starting to think about an actual application, a therapeutic or a product that we might be able to use with this peptide. So that's the first story that I wanted to tell you about, and that's with our antifungal peptide and how we design antifungal peptides in my lab. The second thing that I want to talk about just briefly is a little bit about using peptides to actually deliver things into cells. And these are the students that have worked and are currently working on this project in my lab. So the type of peptide that we're working on in this project is called a cell-penetrating peptide. And cell-penetrating peptides, really, the thing that's most important is they get into cells. So this is an image of a cell here, and the peptides can cross this membrane on the outside of the cell that protects the cell and hold things in. They can cross that membrane and get inside. And what's really cool about these peptides is they don't just get into the cell themselves. If they're attached to something, they can actually carry things into the cell with them. So you can see here, my little ninja is carrying along his box of cargo into the cell with him. And these can be really large things. So these things can be much, much larger than the peptide itself. So we can think about things like DNA, RNA, proteins, even nanoparticles. So these are really large things that normally can't get into the cell on their own. The challenge in thinking about fungal disease is that our knowledge of the interaction of these cell-penetrating peptides with fungal cells is really limited. So my lab has done a lot of work to start to look at how these peptides interact with cells and then how we can design them for improved interactions. So in this slide, all we were doing was taking a set of different peptides, so a set of different ninjas, and we attached a really small molecule to these ninjas. And it's a fluorescein molecule that basically lights up when it gets into the cell. So we can see it by using a microscope. So we can tell if it's inside the cell. And we tested a lot of different peptides. So this is just a few of the ones, those that we tested. We have a lot more. But what we see is that our cells glow with the peptide inside. These bottom sets of images show green, and that green is our peptide. It's getting in. It's not getting into every cell. So if we look at this one on the bottom right, you can see there's only one cell that actually has peptide. Most of the cells don't have a lot. And we've quantified that in this plot. So this is the percentage of cells that actually have our peptide. So you can see that for some of our peptides, all the cells will get the peptide getting delivered. And for some of them, it's not very many. For example, this PEP1, we have very few. So this actually isn't as promising as most of the others. But this is really cool because we didn't know well whether things would get into the fungal cells. They're a lot different than other types of cells, and they have some challenges in terms of their biology. But it turns out these will get in, and we're trying to understand better what lets them get in, and also what can they take with them. You know, in the previous slide, we had this really tiny little molecule. Here, I'm showing now we have this huge molecule. So now you can see this molecule is at least as big as our peptide ninja, but actually even far bigger. It's probably about 10 times as big as the peptide. And we can still get this big protein actually, and this is a green fluorescent protein. We can actually get this into the cells. Our efficiency isn't particularly high. So on the previous slide with our really small little cargo that the peptide was carrying, we were getting about 100% of our cells to have the cargo. In this case now we're down to about 35% or so. So it's not as efficient. But the good news is it is getting in. And so we're trying to think about how we can improve the efficiency and also understand more about what can get in. What's also interesting is for some of these peptides, and in the case of the one I'm showing, the two that I'm showing the results for here, when that gets in, it actually doesn't kill the cells. So sometimes when these peptides get in, they will kill the cells. In this case, we see very little killing activity for the cells. So the peptide itself isn't causing killing, but you could imagine we could attach a molecule that would target the cells and would cause the cells to die. So this might be really helpful in developing new treatments that specifically target the fungal cells, but leave the human cells alone, so we wouldn't have to worry about kind of harmful side effects that we might see from some of our other antifungal drugs. Another thing that I wanted to show you about this is that in addition to doing experiments, which all of the work I've shown you before is done in the lab with living cells, we can also try to model the behavior of these peptides with the cells. And this is work that we've done with Jeff Cloud. And if you remember, I mentioned him on one of those first slides and how his lab does modeling of the interaction of molecules with cells. And so this is a model that's mimicking a cell membrane for a fungal cell. And this is one of our peptides. And you can see we start with a peptide outside of the cell, but as it interacts with the membrane, so this is a membrane that's formed with a lot of lipids. So we have things that have both charge and a very hydrophobic portion. And eventually that peptide is able to interact with that membrane and actually insert itself through the membrane and into the really hydrophobic portion of the membrane. And eventually, if you kept going, this would actually go all the way through the membrane to the other side and be able to access intracellular targets. And we're kind of interested in using these models to now help us think about designing better peptides. So instead of having to do all the experiments, we could actually try to model them first and understand, well, which amino acids in this peptide are interacting. So which one should we change to try to make it interact better? And that's work that actually is ongoing, and we have some initial results that we that we aren't quite ready to talk about, but it looks like we are able to change the interaction by changing those amino acids. So as a quick summary of what I've told you about today, I hope that I've showed you that we can modify peptides that we find in nature or that we design to change their interaction with cells and their effect on cells. I showed you a little bit about how we can design histatin 5, a natural peptide, to resist cutting by fungal enzymes and also improve its antifungal activity against that fungus that I talked about, Candida albicans. We've also showed you that our cell-penetrating peptides can carry cargo molecules, even really large cargo molecules, into fungal cells. So that's what we've done already, but where are we going? I think it's always interesting when you're hearing about research to know what are the next steps. That's actually the most important part, and that's what gets us as researchers excited. So some of the things that we're working on right now is that we're working on trying to engineer our antifungal peptides for not only resistance to fungal enzymes, but also resistance to human and bacterial enzymes as well. Because in your mouth, you actually have a lot of bacteria too. And your own body produces some of those cutting enzymes. So we want to study and try to improve those interactions so that we can really keep the cystatin 5 active for a long time wherever we want to use it as a treatment. We're also working on using our modeling results to design our cell-penetrating peptides for improved delivery, as I mentioned. And kind of as a bigger picture idea of what we're trying to do as a lab, we're really trying to improve our understanding of the rules that dictate how peptides and larger proteins interact with cells and cellular components and other biomolecules. Because if we can understand how these molecules interact with cells, then we can think about engineering them for improved interactions or just different interactions. And I like to think about this as really being this long-term goal of de novo design. So de novo just means designing something from new, so from scratch. So it'd be like, you're taking something, you don't have a recipe, you just know what you want it to taste like in the end, and you're just gonna put things together and it's gonna come out and it's gonna be perfect. So I like to think of this as our peptide vending machine or our protein vending machine. We just go to the vending machine, we tell it what we want. We want it to be stable at a high temperature. We want it to not get cut up by those fungal enzymes. We pay for it, gotta swipe your credit card, and then press the button to get the peptide, and out comes a peptide that does exactly what you want it to do without ever having to do any experiments. So that's really our exciting long-term goal. We're a long ways away from that, but hopefully someday in the lifetime of some of you listening to this, we might actually be able to do that. So before I go to taking questions about the research or other things, and Alpha will also help answer questions, I just want to acknowledge the people who did the work. I already showed pictures of some of them, but what I really want to highlight here is the people in red. The people that I've highlighted in red are actually undergraduate students. And I think it's really important to know that both graduate students and undergraduate students do work in my lab, and they've really contributed substantially to the projects that are going on. And some of these former group members even have already been on papers published in our group. And actually even Alicia was actually a high school student. So once in a while we're able to get high school students in even to help with these things. Also, we need to thank all the people who helped with the work who aren't listed above. And then this is something that you may not know, but a lot of your tax dollars may go to helping support the research in university labs. And it's because research is expensive and we need to get the money from places to help us do it. And so the National Institutes of Health, along with the National Science Foundation, along with the University of Maryland and another organization have helped allow the work that I talked about today to happen. And so with that, I'll turn it back to my vending machine, and also I'll turn it back to Alpha to help us with the questions.
Great, that was an excellent presentation. I definitely was interested in your work on the proteins and the resistance to the enzymes. I hope you guys were as well. And now we'll be open to any questions. I'm looking at the Q&A. I haven't seen any there. So feel free to ask your questions first, but if you guys don't have any, I'll go through a list that I have of questions we could potentially answer. So I'll give you guys like 1 minute. Oh, okay. So Mohammed Sheikh asks, what do the wicks on the bars of the bar charts represent?
Okay, so I'm going to go back to my slides to show you. And I think this is actually a really excellent question. And I skipped this in explaining it, but let me go back to one of them that has some. So the little wicks on the chart, what they do is they're showing the variability of our results. So when we do an experiment in the lab, it doesn't always give us the exact same result. You know, sometimes it's a little bit different. Sometimes it's a little bit higher or a little bit lower in signal. And so these little lines help us to show the level of variability that we have in our results. So you can see in some of our cases, we have very, very small variability. So those results are very reproducible. But in other cases, we see a longer bar. And this indicates that we might expect, if we did this experiment again, that we're going to see the result to be somewhere in this range. It's a pretty large range. And so we have to consider that when we're analyzing our data, because we don't know where the true value, the really, really accurate value lies, because you have to do a huge number of tests to do that. And we're only able to do a limited number of tests. So that's a really good question. I'll also show you another one with the same idea where we can see these little things. And it just shows, if you see small, we call these error bars. And in this case, I've plotted standard error. Sometimes people will plot standard deviation or also confidence intervals. And when you want them to be small. If they're small, then you have confidence that you have the correct value. If they're large, you have less confidence that you have the correct value.
All right, thank you. Hope that answered your question, Mohammed. Before I go into the other ones, just quickly, I want to remind everyone to please complete the voting poll if you haven't already. That information is very valuable to us. Okay, next question we have is Alex Lamb. He says, how much time commitment is research?
Okay, this is a great question. And this varies a lot depending on the lab that you work in. In my research lab, I like to have, for undergraduate students, it's different for graduate students, but for undergraduate students, I like them to commit about 10 hours a week. So it eventually becomes similar to another course. You know, for a course, between the time you're in class and the time you're working outside of class, we expect probably 10 to 12 hours per class. So that's what I look for in the lab. Sometimes when you first start, it's a little bit lower. It's maybe 5 or 6 hours a week. And then some weeks are more, some weeks are less. It depends on the work that you're doing. But usually I look for about 10 hours. Some professors in their labs, they might look for a little bit more, or they might look for a little bit less, but it's usually somewhere around that kind of time commitment.
Cool. All right, the next question is from Ali Hoops. Sorry if I'm mispronouncing your name. They ask, first off, how many hours per week do undergrads typically spend working on these projects? And is it largely a volunteer basis, or do students get pay or credit?
Yeah, and I just talked a little bit about the time commitment. So I'll focus on the payer for credit thing. And this, again, also varies a lot. So typically when students join my lab, I first ask them to volunteer in the lab. And so they're not taking it for credit and they're not getting paid. They're basically doing it kind of like an internship, an unpaid internship, to learn about it and to understand how research works. And then if they do a good job and they like it, in the future, they can take it for credit. So they basically can use it as a course and to replace a course. So in chemical engineering, our undergraduates have to take something called technical electives that are kind of learning about specific areas of chemical engineering. And they have to take 3 of those classes to graduate, and they can count research as one of those classes. So that's kind of a way that they can kind of get some academic credit for doing it. I like to make sure, what I tell my students is, I like to make sure that before I let you take it for credit, you're going to get an A in research. And so that's why I like you to work in the lab and volunteer before you take it for credit. In my lab, normally students aren't doing research for pay. Sometimes there are certain programs that allow that, and sometimes I have funding where I can pay the undergraduates, but in most cases they are volunteering. But again, that's different. There are other labs where especially after you've been in the lab for a while, then you would expect to get to be compensated for it by getting paid. But it does vary a lot. But in most cases, you will be expected to volunteer in the lab before you're going to get paid.
All right, thank you. I hope that answered your question. Next one is another great question by Zhe Zong. They asked, how do we go about, we as in, I'm assuming incoming freshmen or transfer students, how do we go about participating in research, and are there fairs or websites to find these opportunities?
So this is another excellent question. So in order to participate in research, you basically just have to find an advisor. There are sometimes some poster sessions on campus where people have big posters, kind of like a science fair type of poster about their work, and you can learn about what's going on. And there is actually a website that that does exist, and I don't remember the name of it offhand, but we can get it to you if you're interested. That actually helps link students with research programs directly. So faculty can actually advertise their ongoing research projects, and students can go there and look for them. I actually don't advertise on that website because I've found I get enough interest from students who find my website or who have heard me do a guest lecture in a class, and they come to me. So I don't usually advertise in any other way. So that means one of my pieces of advice to you is don't just look at at websites that might already list projects. Go and think about the kind of work that you're interested in. Go to different departments on campus and see which professors are actually doing research that you like. So you can go to my website, for example. Mine's karlsson.umd.edu. You can read through some of the papers from my lab. You can read the descriptions of the work and say, okay, I'm not really interested in medical things. I want to do something else. You could go to Chun-Cheng Wang's lab and see all their work on batteries and be like, okay, batteries is exciting to me. And then you just send them an email and you just say, hey, I was looking at your website, I've read some of your papers, and it looks really, your research looks really, really interesting, and I'm interested in getting involved. Do you have positions open for undergraduates? And so for me, that's the best way, is to meet with professors and talk with them. And don't get discouraged if you hear no, because sometimes we just don't have space. Sometimes we've just taken on some new undergraduates and we don't have space for more. And so sometimes you just come back later. A lot of the students in my lab, when they initially emailed me, there wasn't space, but they emailed me again and they were persistent. And I like to see persistence. Persistence is a really great characteristic of researchers. So when people consistently come to talk to me, I think, okay, this person really is interested in research and they're really interested in my research. And they clearly can stick with things. So I think I'm going to give them a chance.
Definitely, that was great advice. And, no matter what your year is, it's really easy at University of Maryland to find research, especially chemical engineering department. Professors are always willing. So.
Yeah, and Alpha, we can follow up on that too, because I think that was a good point, and I see a question coming up related to this too, that you can get involved at any point. The undergraduates that I have in my lab, some of them started as freshmen, some of them started as juniors. I don't typically let people start as seniors because you have to learn a lot before you can accomplish something. So for me, the ideal time is somewhere between the freshman year and the first semester of the junior year. And we know that if you come in as a freshman, you don't have much experience, and it's going to take longer to train you. But as somebody who's more senior, like a junior, isn't going to be able to stick around the lab as long. So they're not going to be able to work on the project as long. So, so there are advantages and disadvantages to people with different levels of experience and different times. But if you're interested, it's perfectly fine to get started as a freshman.
Yeah, hopefully that answered your question. Terry Goolsby, they asked what year is the earliest a student can work on a research team. As Dr. Karlsson said, it doesn't matter. Ideally, yes, not senior year, at the end, but between freshman and junior year, it will come with its advantages and disadvantages, but ultimately it's still a helpful experience. All right, so we also have a few specific questions. Jason Karlsson asks, you stated that your goal is to make these little ninjas very durable. How long does it take for them to eventually break down? Or do you expect them to be shed from the body?
Yeah, so I think this is a good question and one we haven't specifically explored, but we don't expect them to be around forever. These are made of the natural things that the body is used to breaking down. So what we're hoping is they'll stick around for, probably on the order of a day, maybe a little bit less. We'd actually have to do the studies, but we would expect that after a day in an environment in the body, they would probably be gone. And if they're not, the body would kind of target them to a location that's really good at degrading things like this. One of the advantages of using natural peptides or peptides made from natural amino acids is that the body will eventually be able to break them down, which means you wouldn't see them accumulating. I didn't talk about it today, but there are actually synthetic amino acids. So they're ones the body doesn't make on its own that you make by organic chemistry. And sometimes those have a lot of good characteristics in terms of using them in peptides and in therapeutics. But one of the things that we have to worry about with those is that the body can't break them down. So they could just accumulate in the body and continue to accumulate, and that could cause some kind of toxicity to humans. So one of the advantages of what we're doing, which is using the natural amino acids and trying to make them more protease resistant, is that they still will eventually get torn down.
All right, thank you for the question, Jason. Hopefully that answered your question. And on to the next one comes from Emily Lynn. She asks, how long have you been working on this particular project?
So that's a great question. So I've actually been working on this, let me think for a second. I've been working on the project with the peptides, the histatin 5 peptides, since around 2015, I think, 2015, 2016, somewhere in there. So in about 4 or 5 years or so. The cell penetrating peptides we've been working on a little bit longer. We've been working on those since, I guess, about 2013. But the projects kind of evolve. And that's one of the things that we see in research is that sometimes the projects you start working on or the project you think is going to be really great just doesn't work. You've probably all been at home and thought you had this really great idea to do something and it just didn't work. That happens in the research lab too. So sometimes we just shift directions. Sometimes we go to brand new projects. But these projects specifically, we've been working on for, I guess, 4 to 6 years in terms of the work. And I expect them right now, they're going pretty well and we're kind of generating a lot of different directions. So I expect we'll still be working on these 5 years from now if you come back and ask that same question.
Okay, great. Thank you everyone for your questions. I think that's the last one. Before you guys log off, I just would like to say that we would love to see the poll results now, and I'm going to announce them quickly before you guys have any more questions. So see that there's a lot of incoming Clark School freshmen. Very excited, welcome. Hopefully you guys found this very helpful, and I'm looking forward to having you guys part of the family here at the Clark School. We also have a few incoming transfer students. You guys are also very welcome. Thank you guys for coming as well. I hope you found this helpful before you come to, well, I wouldn't say necessarily campus, but because of COVID, but, being a part of the family, being a part of University of Maryland. And we also have a few faculty and staff. In terms of the major and department, no surprise, a lot of us are chemical engineers. Thank you guys for coming. I know this is obviously our forte, but we also have a few other engineers as well, specifically bio and mechanical engineering. This is very bioengineering focused, so hopefully you guys found this interesting as well. A fair amount of you are not involved in research, so hopefully this was really informative on how you can be if you are interested after this or whatnot. And last but not least, which one is better? 61% said ice cream over milkshakes. I personally love ice cream over milkshakes too.
I feel like this is the most important question this time of year. Yet I'm not sure if I could choose. I like malts. I guess I'm not a big milkshake person, but if you add the malt powder in, then I go for it. But I do like my ice cream.
Yeah. I don't know. I like milkshakes from time to time. I prefer ice. Terry Goldsby asked, how do I take the survey? It should have shown up as you arrived in the meeting with polls, the button polls, but I don't know if it's still there. The polling just ended. I'm sorry about that. But yeah, in the future, I think you just press polls and you take a survey. All right, so we're at the end. Thank you guys for coming. Thank you for all your wonderful questions. I hope you guys enjoyed the presentation. Is there anything you would like to add, Dr. Karlsson, before we go?
No, I just thanks for attending, and hopefully we see a lot of you at Maryland soon.
Very exciting. All right. See you guys. Have a great day and stay safe.
Bye.